is now initiated when the rumen content is high. And the cycle, there are 2 cycles to it. Primary cycle, rumen motility has 2 cycles. And that is what you will deal with tonight. The 2 cycles, you have the primary cycle and the secondary cycle. You can also call it the A sequence. You know, these days it's not easy to do an assignment. It's very easy to do an assignment with the AI. You just find it, and some people will just copy it and dump it on me without even changing one word. You know, when the internet says this occurring in your body, this, they will just copy it and dump it to me like that. You know, you shouldn't do that. You should moderate what you see, what the AI tells you, with your own critical thinking. Paraphrase it, remove some words, bring it to reality in the concept. I mean, around the concept we are discussing, you know, remove what irrelevant sentences, and then that makes more sense and helps you to retain it. But when you just copy and paste it, fine, of course I'll give you good grade, you know, but it's all about learning. It's all about you and not about, you know, me. So when you read these things, yes, try to summarize it in a way that it impacts on your understanding. And then when you submit it, you can discuss it anytime. So we have the primary cycle is also called the A sequence, and the secondary cycle is also called the B sequence. So these 2 control regurgitation. So the cycles, it starts in the reticulum, spreads to the rumen, comes back to the reticulum, and by that process, the material can be regurgitated. So I want you to when you type, just type the cycles of the primary and secondary cycle. Just type the primary and secondary cycles of rumen motility. And I want you to tell me in steps. Put it in order. What happens in each cycle, how it spreads to the second cycle, and eventually it leads to regurgitation. So what goes on in the mouth is not very different from what happens in the oral gastric. So the food is chewed, is swallowed, is retained in the rumen. So this is where the motility comes. So if I were you, I would put here, initiates, the retention initiates what? So this cycle will lead to ruminoreticular motility. So this basically is all about, so you can call it ruminoreticular motility. So that is to say, this cycle involves the rumen and the reticulum. So the rumen and the reticulum, even though they are 2 distinct anatomical structures, they function as a single unit. So that is why in the motility, it's very difficult for us to describe it as rumen motility. It's better to say ruminoreticular motility. So that motility needs to what? Regurgitation. So motility leads to regurgitation. And after that, it goes into the oral cavity back again. And then, so remember that this is what? An antiperistaltic process. The regurgitation is what? Antiperistaltic. So you expect how, look at how the muscles contract, sphincters open and close. So that is what you'll be looking at in that assignment. I mean, have I given you any assignment this semester? Yes. Yeah. The endocrine, have you partitioned the groups are already known?
Yeah.
So you are working on that. Special senses, right? And then reflex. So that's the assignment. Once you do that, you have your 5 points. So please start working on that if you have not started. I'm not sure I will give you another assignment within this semester. So that's just enough, right? So start working on it. And I think a couple of persons have because important. So that's the omasum. So we have the several papillae. These are the leaves. These are the omasal leaves. With each of these having several papillae. So this is now an opening. So this omasum is going to be abomasum. And this is coming from the reticulum. So this is going to be the reticulo-omasal orifice. Reticulo-omasal orifice. And this is going to be the omaso-abomasal orifice. And now this basically creates a canal. This is going to be called omasal canal. So you can see that there is a great curvature and there is a small curvature. So the papillae, this point is the point of great curvature. And this point is the point of lesser curvature. So the omasal canal is formed between the lesser curvature and the omasal leaves. Okay. So basically, the food material is further broken down into smaller pieces here to prepare it for an encounter, to prepare the food to encounter the abomasum, where you have what? Juices, gastric juice that will now further embark on chemical or enzymatic breakdown. So abomasum in cattle is quite large, in sheep is short, it's quite small. So the omasal canal is a short canal, it is not long. A short canal formed by the lesser curvature and the omasal leaves. So those can be referred to as many plies of the leaves. No enzymes are formed. The basic function is to crush the food further and then absorb water and volatile fatty acids. So at the end, the material will be, if you look at the material in the reticulum, it is dry but still a bit rough. But in the omasum, it is as well dry and then very fine particles. So although dry, the fact that it is now in very finer particles. So the medulla is considered the gastric center. The medulla oblongata is considered the gastric center. So that is where gut motility is controlled. So if that is the medulla, it's divided into the left and the right. So this is the left side, and this is the left side, and this is the right side. So the nerve, the vagus nerve, come down from there. So when it leaves the left and the right, it gets to the thoracic cavity. So this is the medulla. This is the thoracic cavity. So when it descends to the when these nerves come out from the brain, from the left, and they get to the thoracic region, it divides into two. This is the dorsal branch of the right vagal. This is now the since it is the vagus nerve, this is the vagus nerve, this is the vagus nerve. So this is going to be the left vagal nuclei, right vagal nuclei of the medulla oblongata, right? So this is going to be the nuclei. It contains several cells. The same thing, so this is going to be the right vagal nuclei of the medulla and the right. So the vagus nerve from the right and the left comes down towards the thoracic region, divides into two. This is going to be the dorsal branch of the right vagus nerve, and this is the ventral branch. The same thing goes for this. This is the dorsal branch, and this is the ventral branch. So the two dorsal branches, they unite. The two dorsal branches unite, and then the two ventral branches unite. So this is now going to be called the dorsal trunk. This is now the dorsal vagal trunk. This is now the ventral vagal trunk. So, and this is now the stomach. This is the rumen. The rumen is divided into the dorsal section and the ventral section. Remember that the dorsal section is divided into the cranial and caudal. So now the dorsal vagal trunk will supply everywhere, while the ventral trunk supplies only the ventral. So this supplies only the ventral, this supplies here, here, here. So the vagal motor parasympathetic supply to all regions. The vagal motor parasympathetic supply to all regions. So that's what we have said there. And then the dorsal vagal trunk, the supply to all regions of the lungs is by what? The dorsal vagal trunk, which is formed by what? The dorsal branch of the right and left vagal. Does that make sense? Mm-hmm. Is this speaking the diagram? Good. And then the left and right vagal, of course they come from there. Then the ventral vagal trunk formed by the ventral branches supply all parts of the stomach except the dorsal part. Does that make sense? So that is why. So the dorsal vagal trunk supplies all parts of the liver. So that is just repeating what you have here. So that diagram speaks to the content of the slide. So what is the time now? Okay, so we can take a break and come back at 2:10. Manipulate, you can engineer the rumen by changing the population of either the protozoa or the so if you have defaunation, it means you can also have what? Faunation. So defaunation is the removal of the microbes, especially protozoa. Faunation is to do what? Introduce. You know about flora and fauna, right? So we use what we call rumen fistulation technique. You can create a rumen fistula, open up the animal, implant the rumen fistula, and then do whatever you want over time. You can even remove it and put your hand into the rumen, collect rumen fluid, close it again. So you can watch it on the YouTube. Rumen fistulation techniques. I did it severally in West African dwarf sheep. All you need to do is to open up the rumen and implant your rumen fistula. Put the clamp, put the rubber up and close it. So whenever you want to have access to the rumen, and the animal can live as long as you can manage the trauma, so that there will be complete asepsis. You don't want infection to take place, so it will just be there. But in cattle, being a large animal, you can carry it for years if all the conditions are met, and you can have access to the rumen. And so you want to study the rumen microbial population based on certain conditions. Just simulate the condition and then have access to the rumen and do your studies. So this must be, so the rumen population, the bacterial and protozoa population must be balanced. Again, there is this competition between them. The protozoa tends to feed on bacteria and control their population, which is good. So that competition, just like in any other ecosystem, do exist. But under normal health conditions, the competition is healthy to maintain the population of the microbes. Acidity of the environment kills the microbes. So it is important that the pH of the rumen be maintained. And this is supported by the amount of saliva that we talked about, the salivary secretion, all of that. It has also been shown that shifts in diet affect the microbial population. You don't want to introduce new diets to ruminants instantly. No, because the ruminant animal thrives on the presence of the microbes. So if you do anything that will change the population of the microbes drastically, it is going to affect